the Dementia Research Institute node here at Edinburgh is quite something. And it's worth pointing out its success, which has been substantial and will continue to a large extent, is because of you. Because when it kicked off 2015-16, it was a motley crew of 5 people led by you, with Tara, Joanna, myself, and Barry. You've steadfastly developed and grown, and you've grown it by a focus on scientific excellence and identified a niche, which is neurogliovascular, and then from that, everything else has emerged. So we owe you a debt as well. I want to now welcome yourself, George. George, I've got to tell you, because of you, this room is much more full than it would normally be. And not only by people who are the usual suspects, colleagues from the Dementia Research Institute, there's a very decent turnout from eminent other researchers from the vascular space, the virology space, the intensive care space. We've even got people who come from Dundee to hear this. So that alone says something about you. For those of you who don't know George, a couple of things. He's not somebody for ceremony, so I'm not going to talk much about his academic accomplishments. That would be a lecture in itself. Suffice to say, he recently, just a couple of months, was it last year, the MRC Millennium Medal. George was, and there've been a very small number of people who won the MRC Millennium Medal Award. That is a very, very major achievement, and you'll hear why. I got to know George though just over the last 2 years. First time we met was in a pub in Bangalore. I was with Andrew Morris, and we were walking. We went into a pub in Bangalore, and then Andrew met you. I knew of you by reputation, and then you were with your wife Caroline. Well, we'll go into all the details, but Caroline's got an identical twin who I know very well. So I thought Caroline was Jane Rhodes, who I worked with in a different walk of life. So it was a completely mad moment. And that's how we met. And then we bonded over watching the cricket in Bristol, the women's T20, India versus England. What shocked me, George, as an Englishman, are you from Yorkshire or Lancashire?
I was born in Lancashire, but they made it Cheshire.
Okay, but for a Lancashireman, you've never been to a cricket match, which I found astonishing. So that's how I got to know George. And then since then, you've been fantastically helpful to me around various research areas, some linked to the DRI and others not. I've got to, in this introduction, read out George's biography. I don't know if any of you have read this. Right? This is quite extraordinary because George, as you know from his scientific achievements, is in many ways, you're a national treasure. You're up there with David Attenborough. Right? But just listen to this biography, it's brilliant. Now, who knew this? You were a member of the noise terrorism outfit Scum Auxiliary in the early 1980s. So that was some sort of band. and you're very honest. Artistic and commercial success eluded you. So you had to survive, and you have survived, barely thrived as an epidemiologist. Okay? You also did medicine, and I think there was a risk that at some point you were going to be a neurologist. Yeah, I mean, neurology would have been better off, but I think the field of science and advancements would have been a lot worse off if you had drifted in neurology. You'd end up like me or half a dozen people there. So we're very delighted that you're here. We're delighted that you, I don't know if you would give us a rendition of Scum Auxiliary, but you're going to tell us something about Mendelian randomization. But more than anything, thank you very much for making the trial to come. Thank you.
Thanks very much for that very kind introduction. So I'm pleased that people who aren't from the DRI are here, because I'm going to talk about Mendelian randomization in general, and I'm going to talk about the principles of it, not about applications to dementia at all. So I'm going to say that Mendelian randomization uses the special properties that germline genetic variants have to help understand how one phenotype influences another phenotype. So it's entirely about phenotypic causation. And the motivation for its introduction was that in the late part of the last century, there was a large number of randomized trials that appeared that were testing out hypotheses that were generated by large-scale prospective observational cohorts studies. And one after the other, they showed that things that were thought to bring from cohort studies to be protective, like a whole host of antioxidant vitamins like selenium for prostate cancer, or HDL cholesterol was supposed to protect against coronary heart disease. All of these trials failed. And there's just obviously something wrong with the approach of just observing things, adjusting statistically for confounders. They were getting wrong answers. And it's intended to improve causal inference regarding the effects of interventions aimed at improving population health, and was not at all about genes for X or personalized events. So what it was saying that in the epidemiological setup, which had got these answers that were wrong, you would just measure a modifiable exposure. You would relate it over prospectively to an outcome like coronary heart disease, but there could be confounding factors that influence both the exposure and the outcome. And reverse causation, which is a particularly insidious form of confounding when early stages of the disease process influence the exposure. And you simply cannot adjust for those statistically, as I'll come to later. But if you took a genetic proxy for the exposure, but like an LDL protein receptor mutations that in FH were related to LDL cholesterol level, then there would be no confounding, because conventional confounders, behaviors, socioeconomic factors, etc., can't influence the germline genetic variant you're born with, and there can be no reverse causation, obviously. So in that sense, you can just combine the association between the genetic variant and the outcome, and basically divide it by how the genetic proxy relates to the exposure, and get some sort of estimate of the effect of the modifiable exposure on the outcome. So it's a very simple setup. And the justifications for using germline genetic variation for looking at how one phenotype influences the other, the justification for that, which I'm going to go through in some detail, comes from natural selection. So in natural selection, you basically have it's a phenotype. The selection in natural selection is of the phenotype which the genetic variants might influence, but it's the phenotype which leads to the selective advantage, and the phenotype drags the genetic variant along with it. And Darwin used the notion of the discussion of longer legs being able to get away from predators, for example, and also sexual selection of factors that made animals more attractive to mates. And here's peacock, and of course the DNA of the peacock influences its display, and the display influences its the fecundity, the number of offspring it has. But here you have one phenotype, which is the display, influencing the outcome, the other phenotype. So natural selection is entirely about phenotypic causation, and in fact, natural selection is a sub-component of Mendelian randomization. So Mendel, you all know, Mendel. Firstly, he understood the fact about sample sizes, that you needed vast numbers. Darwin actually did a cross of primrose where he got a 3-to-1 ratio, but the numbers he did was really small, and it didn't stand out, and he didn't carry on doing studies in larger numbers when it would have become clear that the ratio was stable. So firstly, he understood what it took years for many other scientists to understand the need for very large sample sizes. But then he talked about that as just mere chance, just sampling variation. Then he talked about pure chance, which is, in the case of his two laws, that it's purely a matter of chance, which of the two the A or the A will unite equally often with each A or a big A or little a n. So that's the first law, the law of segregation, and the second law, which is the factorial law, is that the traits are independently distributed with each other. So there's two sort of forms of randomization that meiosis produces. Obviously, physical linkage and then the population level linkage disequilibrium. There's partly it goes against that, but as a general phenomenon, it's true. So those were his two laws, and that's what the name is. Mendelian randomization, his randomization, and Mendel's randomness leads to Hardy-Weinberg equilibrium. These are sort of the null model for a lot of population genetic phenomenon. Mendelian expectations are tested against the null, many analyses. That also, this is the Markov process. We've got Goldman had this notion of ancestral heredity, which in terms of predicting phenotypes work perfectly well, but the actual genetic process is purely Markovian. It doesn't matter the generations before your parents. Doesn't influence how the gene will express itself in you. And indeed, this hidden Markov models before Markov, like Haldane in 1919, were introduced because it is such a clear and obvious Markovian process. This also allows for directly estimated heritability, but it's also the reason why people are sitting here listening to a talk about Mendel's randomness, is that it prevents genetic cheating. And if there was genetic cheating, there would not have been the development of complex life. And there's a brilliant book by Mark Ridley, it's not Matt Ridley, no, Mark Ridley, on this, about how meiosis avoids the fact that forms of genetic cheating would otherwise occur, and I think it's got vitiate the development of complex life. And it provides the variation for selection and fair tests, because it's a perfect process of the random distribution of the variance. And then you actually have trials, if you like, of these variants, and which ones improve or hinder fitness. And R.A. Fisher, discussed the only time he ever talked to both statisticians and geneticists. He said genetics is peculiarly flavoured because because of this randomization, the geneticists, even if they wanted to, can't distort their data. That was Mendel's first law, that randomness. And then secondly, the factorial nature of these traits is mimicked by the factorial randomization that was introduced of different treatments in trials at that stage. And his daughter, Joan Fisher Fox, wrote a piece that I asked her to write 15 years ago, about how it was actually the Mendelian processes which gave Fisher the ideas about randomization. So Mendelian randomization actually came before experimental randomization that, of course, Fisher introduced. The other genius, and much nicer genius than Fisher, who was a loathsome eugenicist, the nice genius is Cyril Wright, who was not a eugenicist at all. Wright, a few people here might recognise this, that he read his father's maths books and learned to extract cube roots before entering school, a skill that he said brought him instant lasting unpopularity with the other students. And I absolutely love this picture because this is his second path analysis, and there's this chance, with the germ cells. Chance is actually put into his figure, and it was this chance which allowed you to actually get causal causal relationships out of observational data. But he said that this is just because you know that the crosses he was doing, obviously not molecular genetic anchors, but those crosses anchored the data and allowed you to draw ordered or pointed paths where you knew that one factor was causing another. And I'll only talk about all of his analyses, by 2 or 3 were anchored by the crossing of the genetic crosses that he did. And one of them, he examined sort of mating in non-humans and demonstrated it's through phenotype. And a paper using Mendelian randomization by Lawrence Howe using UK Biobank data, where there's spousal pairs, demonstrates that the fact that there is a sort of mating by alcohol consumption is actually due to the alcohol consumption. So you can work out what are the selective processes, what actually causes those things. And when Mendelian randomization was introduced strictly under Mendelian principles, and it was said that the true Mendelian randomization was within families. Now that is true randomization, that myopic randomization, and that is how the studies should be done when it's possible to do them. But at the time, in 2003, there was virtually no data within families. So that's how we should it within families, that's how it should be. And so we referred to approximate Mendelian randomization using population data. And of course, there can be population stratification, there can be factors which distort things. And we said, we should have stuck to the name approximate Mendelian randomization. This was before there was antimicrobial resistance, so we could have got that abbreviation, because the terrible Mendelian randomization papers that appeared would have been abolished if people had been suspicious of those. And it's not instrumental variable analysis. It is not merely instrumental variable analysis. You're just using the properties of genetic variants to actually help you on fertilization. Because there was many examples early on in Mendelian randomization where the genetic variants did not relate to the exposure, but they still allowed you to actually infer causality with respect to phenotypes. So I like this one very much. This was about sheep dipping, and there was a health, fatigue effects, depression effects that related to using sheep dip. Was this actually caused by the sheep dip, or was it by permissible occupation or whatever? And this beautiful study by Nicola Cherry looked at peroxinase, a functional variant in peroxinase, which detoxified the sheep dip. And it just looked at people who used sheep dip, and it showed that those who had the version which was detoxifying it were less likely to get any of the symptoms. So that was very strong evidence that the symptoms were actually, that the things were actually caused by the sheep dip. But the peroxinase genetic variant did not relate to whether you got exposed to sheep dip or not. It was unrelated to that, so it was not an instrument for the exposure, but it still told you about the outcome. Or in another case, you looked at this is on whether smoking caused bladder cancer. You looked at the variant of slow acetylation genotype, and acetylation sort of clears some of the particular carcinogens in tobacco smoke. And if you were a smoker, if you'd ever smoked, then if you had this slow acetylation variant, you were more likely to get bladder cancer. But you have the beautiful negative control of the fact that if you've never been a smoker, this variant didn't relate to bladder cancer at all. But again, the variant does not relate to smoking itself, so it's not an instrumental variable for smoking. But Mendelian randomization currently is largely implemented now with the instrumental variable framework, such that the genetic variant Z is seen to be the instrumental variable, which has to reliably relate to your exposure of interest in an ID framework. Now, there should be no confounding of the genetic variant and your outcome. Now, there's obviously not conventional confounding, as I discussed, and that is generally the case. But there is possible confounding, because one thing which has a causal relationship to your genotype is your parents' genotype. And now the parental genotype causes your genotype, and there might be some pathways to your outcome through the parental genotype, which is not through your genotype. And this is the only example we could get of family-based data. We use family-based data for making estimates of the effect of folate, maternal folate during pregnancy on neural tube defects in the offspring. And you see, if the mothers carried the genetic variant in the -677 MTHFR variant, which is related to lower folate activity, then the offspring were twice as likely to have neural tube defects if they were homozygous for that variant. Again, you have a beautiful negative control in that the father's genotype obviously does not influence the intrauterine environment of the developing fetus, and the father's genotype is essentially unrelated. But the fetal genotype, of course, as it's dependent on both parents, does relate to the outcome of the neural tube defect, but to a lesser extent. But if you just had the experiment, if you just had the data on the offspring, you'd be misled to think that it's the offspring's folate which was influencing the outcome, when it was actually the intrauterine folate, and that's how the folate supplementation in randomized trials does indeed substantially reduce the risk of neural tube defect. So those are called dynastic effects, when the parental genotype can influence the offspring, but not through the offspring genotype. And if you have genotypes related to alcohol consumption or smoking or whatever, you would grow up in an environment where there's more alcohol around, there's more smoking around, etc. And so you can get confounding, but it is generated by the dynastic effect. So that was that's the second IV assumption. The third IV assumption is that the effect of the genetic variant has no influence on the outcome, except that is mediated through the exposure of interest. So there's no horizontal pleiotropy, as it's called in the genetic context. The economists refer to this as the exclusion restriction, not particularly helpful term. And when you read it in the economics presentation, they often say that Z was independent of Y, conditional on both X and C, i.e. the confounder. And that's a sort of worrying thing, because the whole point of doing Mendelian randomization is to avoid confounding. And the reason why that is true is important and often overlooked. A lot of early MR studies, people would say that they would adjust for the exposure, say body mass index, and then they would say that the genetic variant still relates to the outcome even if you adjust for the exposure. So there must be some other effect. It must be working in other ways. So there's two things. Obviously, you have measurement error in the exposure, which means you're not going to adjust for that fully. But secondly, if you adjust for the exposure, you can introduce confounding or form of bias when none exists, and that would be collider bias. And it's a sort of important concept and possibly under recognized, and that is that when you stratify or adjust for something, you can generate associations between things which don't exist in the base population. You generate bias where none exists. So imagine a sort of US college where students are admitted to the college either through getting an academic scholarship or getting a sporting scholarship. And then you think that in the general population, there's no association at all between academic ability and sporting prowess, none at all. But if you go onto a campus of one of these colleges, and you throw a ball at one of the students, and the student tries to catch it, falls over backwards, and the ball bounces off his head, you can confidently go up to them and say, "You are very clever," because there's no way that they are there because of a sporting scholarship. So something which is unrelated in the population, once you stratify or adjust for some factors, can become strongly associated, actually minus 0.6 correlation in that case. So when people just sort of think adjusting for things is really good, stratifying is really good, you can actually introduce serious amounts of bias by doing that, by adjusting, and that's referred to as collider bias. So horizontal plotropy, genes always operate through a phenotype, so it must be working through some unmeasured phenotype. But that obviously, if you have if the genetic variant is having two effects and two entirely independent pathways, then that's a serious problem for Mendelian randomization, which depends on the fact that you're saying that all the effects are through the phenotype that you know. Now, plotropy comes in sort of two forms. That's horizontal plotropy. The other form would be vertical plotropy, and that is when the genetic variant influences one trait, and that influences downstream trait, like a genetic variant influences BMI, and BMI influences blood pressure. That's vertical plotropy. But you'll notice that vertical plotropy is Mendelian randomization. So Mendelian randomization is simply exploiting vertical plotropy, which are downstream chains of phenotype. Now, in some cases, you might have a situation, or very often you will have a situation where you don't know how the genetic variant is influencing your exposure of interest. Now, that's fine as long as whatever that upstream factor is, so that's some sort of molecular phenotype, as long as that it isn't influencing your outcome. It might mean that the actual primary phenotype is you, but you don't know what you is, but if you isn't influencing the outcome, that doesn't matter. But if this upstream factor does influence the outcome, then you get the worst form of bias, which is correlated plotropy, because nearly all of the sensitivity analyses for Mendelian randomization can't deal with this. There are a few that can, but most can't deal with the situation where you have correlated plotropy, where the effect through the upstream phenotype correlates perfectly with the effect through the phenotype that you think you're studying. So the fundamental assumption of Mendelian randomization is gene environment equivalence, which is seen, for example, in many Mendelian situations, Mendelian disease situations, such as considering pellagra as a condition which is produced by niacin deficiency, dietary niacin deficiency, but the same phenotype is produced by a condition called Hartnup disease, which would be the differential diagnosis of pellagra. It looks like pellagra, but Hartnup's this disease is caused by a genetic influence on amino acid transport, tryptophan transport, which was converted to niacin, and the downstream effects are then the same. The two things look the same, whether you're on low niacin is due to genetic change or is due to dietary deficiency, it produces the same downstream phenotype. So this is just known as like in genetics, you have famously, Goldstein introduced the term phenocopy, which is that the environment, you can have environmental change which copies what genes do. In the Soviet Union, where they were more concerned about the environment than genes, Schmalhausen called it genocopy, that the genes could copy what the environment does. But they both are saying that the genetic and environmental effects can be equivalent, and it's assumed by the Breeders function. It's a sort of quote, no doubt all environmental effects can be mimicked by one or several mutations, and I think that's true for endogenous disease processes. Possibly being hit by a truck, you can't actually think of genetic mimic, but for endogenous disease processes, and it's in developmental genetics with this gene environment equivalence term is used. So if you think of the maternal folate example, the exposure, the environmental exposure is maternal folate levels. You're looking at offspring or acute defects, the genetic perturbations the variant I discussed, the environmental perturbation you want to think is equivalent is dietary folate acid deficiency, and as discussed, the actual mechanistically, these two work in the same way. The dose equivalence is reasonable. The -677 variant has quite a large effect on folate activity. Timing equivalence is this is all happening during pregnancy, so it's happening at the same time. So that's equivalent. So this sort of overall, this sort of strong gene environment equivalence in this situation. All the LDL receptor variants, LDL receptor mutations, which cause familial hypercholesterolemia. The heterozygotes with familial hypercholesterolemia have very considerably raised LDL cholesterol, considerably raised levels of heart disease. Environmental perturbation will be cholesterol-lowering medication, like the statins work through upregulating LDL receptors, which then means cholesterol is taken to cells. So mechanistically, they're equivalent. The dose equivalence is similar. Similar differences or even larger differences in cholesterol, seen with genetic variants in some cases. But the timing equivalence is only partial because the genetic variants lead to a difference in cholesterol right from birth to on, and atherosclerosis is developing right the way across life, but you're only getting your statins when you're 50. So there's not equivalence, there's not dose equivalence, and there's not timing equivalence in that case. So the plausibility is sort of reasonable, but you have to take into account this difference in timing. Cholesterol is having effects in early life, particularly, for example, when it wouldn't be the case that there was that equivalence. So you need to have additional knowledge to say whether these things are reasonable. I mean, I think gene environment equivalence is shown very nicely by the fact you've got a whole host of cholesterol-lowering drugs which target specific genes. So you have HMG-CoA reductase, the gene, which is inhibited by statins, and then you have the same picture, or PCSK9 inhibitors. There's PCSK9 genetic variants. There's drugs, PCSK9 inhibitors, which inhibit PCSK9. And so that naming equivalence is rather convincing. Now, it's behavioural factors like smoking, heaviness, there's genetic variants, and the nicotine receptor gene, which leads to people smoking more heavily if they smoke. So it doesn't actually influence smoking uptake, doesn't influence trying smoking, but if you do smoke, you smoke more heavily, you get more nasty stuff, you have higher rates of lung cancer, heart attacks, etc. And again, mechanistically, these are equivalent. There's a lovely negative control here, because if you look in people who've never ever smoked, you find the genetic variant doesn't relate to any of these nasty things. So these are lovely situations when you have these interactions. The most convincing MR cases, in my view. There's dose equivalence on this time equivalent. So those are situations where gene environment equivalence is reasonable. So, sort of pin-up example early on in Mendelian randomization was C-reactive protein, which is an inflammatory factor, which anything bad that happens to you puts up your C-reactive protein. At the end of the 20th century, it was started being called the new cholesterol. Pharmaceutical industries were developing drugs to lower C-reactive protein because of the very strong observational associations between C-reactive protein and high risk of coronary heart disease. Now, they adjusted for a vast number of factors, and that, it attenuated a bit, but it stayed massively significant. Some people like to call it findings. And these are all the confounders that were adjusted for in this analysis. So, a lot of confounders were adjusted for, and you notice the confounders relate very strongly to C-reactive protein. Everything bad relates to higher C-reactive protein, like smoking, obesity, etc. So you can see that this is massively confounding. With a variant, a promoter region variant, cis variant in CRP gene, which relates to higher CRP levels, you see that the only thing that relates to is CRP levels. Mendelian randomization does what it says on the tin in situations like this. And unsurprisingly, I'm telling the story, it's when you did the Mendelian randomization of coronary heart disease, there's none. Now, that meant the pharmaceutical industry, because of MR studies, stopped trying to develop these drugs. That was about to save themselves huge amounts of money. But actually, the most important thing on this slide to me is this result. This is what a completely non-causal association looks like after people have adjusted for a vast number of confounders. That is, adjusting for confounders is very often almost useless. And the fact that papers appear every week saying that coffee causes this or prevents this or whatever, and that basically, if you look at the results, and if you see something which looks like that, then you should think that is precisely what you've seen, precisely what you should expect with underlying null association. So you can use Mendelian randomization to find out what the confounders are. A very likely confounder with the CRP, amongst the many, is body mass index, and Nick Timpson, who have been involved in identifying FTO as the first common variant related to a reasonable difference in body mass index, used MR to show that BMI does indeed put your CRP up. But the point is that that means that FTO does indeed relate to CRP. And that is a perfect example of what I refer to as correlated polytropy, because any genetic variant which relates to BMI, when you get a large number of sample sizes, will relate to CRP. And FTO is about the fourth strongest genetic variant related to CRP, in fact. And of course, BMI influences coronary heart disease. So if you just take all the variants, then including all the BMI variants, and you just get the wrong answer. But you can co-ops or Steiger filtering, which is to look at the variance and see the variance in the exposure explained by the genetic variant should be considerably greater than the variance or pseudo variance of the genetic variant on the outcome. And that it's very difficult to come up with a situation where that isn't the case. That almost has to be the case. And so you can use this to filter out things which appear very obviously to be inappropriate. So the reverse direction CRP didn't have an observation associated with BMI, obviously, but it was thought that CRP, some people were saying CRP actually increased obesity at this time. But CRP has no effect on BMI. And if you consider gene environment equivalence in this situation for body mass index, FTO relates to higher calorie intake in Alsbang, and that's reasonable sort of gene environment equivalence, because that's the sort of thing you might want to do to check to reduce obesity. Physical activity level, that would be reasonable. I mean, Miguel Hernan, causal inference person, he often makes this joke about the fact that if you want to lower your BMI, you could just chop off your leg. That would actually be very effective. Your BMI would go down substantially, probably even more than taking GLP-1 agonists. But chopping off your leg is unlikely to reduce your coronary heart disease. And 25 is quite significant smoking-related variant for CRP, and obviously that would be rather suspect as taking that as something which would be the equivalent. And for CRP, cis variant in the CRP gene, which relates to higher level of CRP, that's reasonable. But the FTO genetic variant obviously is not the equivalent, nor the IL6R genetic variant, which regulates for CRP, regulates CRP and influences lots of outcomes, or the smoking variant. And anonymous genetic variants are often, which are often used in Mendelian randomization, would be suspect. And as GWAS's genome-wide association studies get larger and larger and larger, more and more of the hits will be for upstream factors, often very far upstream factors. The further upstream you are, the more likely it is that something in that upstream chain will be influencing your outcome. So larger and larger GWASs are not actually necessarily helpful. And there's the does what it says on the tin phenomenon. If you now use all the genetic variants that have been found for CRP, and you do naive Mendelian randomization, it appears CRP causes everything. Because, of course, this is now the genetic instrument of putting all of those variants together. It just relates to the confounders. So you just bring back all the confounding in, you just naively use all of the genetic variants. So a nice example, I think, demonstrating various many of these things, is to look at sclerostin. So sclerostin is an exemplar of a genetically informed therapeutic target, is sclerosis and Van Buchen's disease, both conditions where people have literally no sclerostin and extremely high bone mineral density. And it was because of that that monoclonal antibodies against sclerostin were developed, and they work extremely well in increasing bone mineral density and reducing fracture risk. So we did a GWAS of circulating sclerostin levels. This is sclerostin in the blood. Found two variants which weren't cis variants in the source, but they had reasonable biological support in terms of how they influence the sclerostin levels. And you do MR with that, and then indeed having higher sclerostin levels is related to greater risk of fracture and higher bone mineral density. So all that would look good. But then you do a larger GWAS, sort of like the GWASs get bigger and bigger and bigger. And this time now with a quite large effect, but low allele frequency cis variant and SOS in sclerostin, the gene itself, would like to be by the instrument found. But a fair number of these upstream these other variants actually influence bone mineral density, and then through bone mineral density influence sclerostin, because biology is homeostatic, and stability. It's basically stability through micro changes, through fluctuations, and this is MR. This is MR just showing that bone mineral density does indeed influence sclerostin. Higher bone mineral density will lead to higher levels of sclerostin. And this, of course, will give you the exact reverse of the answer. It will give you the completely wrong answer, not just null. It would flip the answers. If you're taking genetic variants that relate to bone mineral density, and through that relate to increased sclerostin levels, you would get exactly the wrong answer. So these are the sorts of analyses that have been done, just throwing all the genetic variants in and getting published in Mendelian randomization today. Now, the appropriate cell or tissue type for the phenotype, we're talking about sclerostin here, would obviously be sclerostin where it's produced in the bone by the osteocyte derived cells. And what you're looking at in the blood is just what's sort of leaked out. It's not sclerostin in the blood which is going to influence your bone mineral density and fracture risk. It's got to be taken up by the bone, but not taken up. It's actually produced in the bone locally. And the SOS variant, when you can do Mendelian randomization using that cis variant, you get a much larger effect on bone mineral density because for the similar influence on the leaked out sclerostin which is circulating in the blood, which is just the wrong tissue for this. You want to know the sclerostin level in the bone. So we're often using phenotypes which are just measured in the wrong place. We take phenotypes because they're conveniently available. And one's got to think of what is the actual appropriate tissue and the appropriate cell type that you should be looking at. So MR, what it is. So a lot of studies are two-sample MR studies. I call these noodles for reasons I'll come on to. They use summary data for both the exposure and the outcome. And these are the ones where you can do an MR study in 10 minutes with using summary data. A lot of the early studies was one sample MR studies where you both derive your instruments and apply them to the outcome. Then that's fine, except there is a problem if you just get overfitting when you're GWASing both the exposure and the outcome in the same sample. You get overfitting Wiener's curves. So what you need, if you're doing that, for example, working with biobank, and that's the only large sample that has the exposure and outcome data you want, then you can use a jackknife approach whereby you split the sample into 10 tenths. You combine 9 of them to 90%, you GWAS them for the exposure, you then apply them to the other 10 to generate an outcome effect, and you do that 10 times, and then you meta-analyze them, and then you avoid Wiener's curves. Or very often now there's a hybrid design where you take external GWASs for your exposure and you apply it often to UK Biobank as the outcome, the individual level outcome data, and you don't have that, you don't have the overfitting problem. UK Biobank or all of us and you don't have the other, you don't have the other fourth place, the unicorn. But how MR is really, in 2016, 2-sample Mendelian randomization started overtaking 1 sample. And this is Fernando Hartley wrote a critique of how 2-sample Mendelian randomization studies were being done, demonstrated problems in one particular study. I mean, you could have done this for large numbers of studies. And Fernando's paper led to that study being retracted. Now, you can't critique each one of these papers, but thousands, tens of thousands of MR papers should be retracted because there were mistakes in what they've done if you looked at them closely. And this is the MR papers as they're appearing. This is actually a true exponential. In about 3.5 years' time, every single paper in the world will be a Mendelian randomization paper if this carries on. And so UK Biobank has data on breakfast skipping, so you can GWAS breakfast skipping and you can look at it against every single fucking outcome in the world, and you can publish the paper. And even when people do systematic reviews, they say about the Mendelian randomization studies, you know, RCTs, they're the important ones, and reinforcing potential causal links. But it's just nonsense. So once you think of gene environment equivalence, it becomes very clear that these things are nonsense. It's very unlikely that it's sort of, and of your 28,000 genes or whatever, it's very unlikely that any sort of specific genetic effect on breakfast skipping, which isn't working through some upstream effect. And then when you look at all the variants that do relate to breakfast skipping, relate to smoking, you know, et cetera, and all the things you'd imagine. So it's just ridiculous. And everyone just considered, is gene environment equivalence, is that at all plausible in this setting? And you realise very quickly that the answer is no. There's many sensitivity analyses for Mendelian randomization. There's an industry in generating them. This is the template for these papers. You say current methods of limitations. We simulate data and apply our new method and handful of previous methods to these, but our method performs best. We are the champions. That's a method thing. So noodles, no moles, numbskulls. So noodles this is unfair really on this paper, because apart from the fact it was ridiculous to think you could actually get specific variants related to noodles, it was well done. In other respects, it was a well done study. It's just that it just fails at the plausibility at the gene environment equivalence level. But anyway, I started calling them noodles after that. And these are studies I won't waste my time. This is from a website where you're advertising writing a Mendelian paper, randomization paper for you, basically. And they're getting the side. They say, you know, we've got into a journal with an impact factor of 50. We can get a paper for you into. These are from paper mills in China. There's a price list, 3,398 yen per paper, and you get 5 for the price of. You get 5 for the price of 4 if you buy a package. So then when a new exposure comes out, a new GWAS comes out, it's amazing how suddenly loads of people are interested in exactly the same thing. The causal effects of gut microbiota on the risk of erectile dysfunction. Exactly the same data sets, same things, all you know, apparently different, you know, supposedly different authors, all sold by different paper mills to people. So that's the reality, and that's why there's this. That's why the takeoff, and that's why most of the papers are mostly terrible. There's now no nulls. The Mendelian randomization studies that really mattered were things like showing a lot of them were showing nulls. You know, most things aren't caused. You know, show HDL cholesterol wasn't protected. CRP was unlikely, didn't influence the risk of coronary heart disease, and actually many others which actually could have stopped some drugs being used. There's no null papers. I mean, who's going to pay for a null paper? And then numbskulls. These are methods which are so complex that the people who develop them don't really understand them, but they are rushed out and utilised. And an example, a very important null for vitamin D. I mean, in Mendelian randomization, apart from multiple sclerosis, where vitamin D there is a protective effect of vitamin D on multiple sclerosis. Everything else, it was not, coronary heart disease, diabetes. Observation of vitamin D has been said to relate to all of these things, but the Mendelian randomization for these in general populations is not. But then, a non-linear method was brought out, which I haven't got time to go through precisely why it produces nonsense results. But these are the observational associations of vitamin D, very high rates of everything, and then at about the median population level, it levels off. When you get an exposure which relates in the same way to a load of outcomes, you get very suspicious because that's not how biology works. That's how confounding works when we've got vast number of confounders which influence many things. So they did this. They used a specified approach, and they then said that they showed that suddenly there is a protective effect of vitamin D, even though all the standard conventional MRs show there's no protective effect. And this got picked up by all the vitamin D enthusiasts and was actually quoted by guidelines. And 100 authors, some of the most well-known cardiovascular and metabolic researchers in the world, 100 authors apparently looked at this figure in the paper and thought it didn't ring any bells to them. That you had 4 strata, and you did the MR, and it was sort of protective in all 4 strata lay to the left and substantially to the left of the overall effect. The overall effect's null, but every strata shows benefit. Now, sadly, people have this vague memory, been taught about Simpson's paradox, and doesn't Simpson's paradox mean this can happen? No, not when you've got a causal interpretation of the effects and you know the distribution of the variables. This is absolutely impossible. It's just ludicrous. In fact, it is ludicrous. So you just recapitulate these long answers. And a negative control will be that you can instantly, you use exactly the method that's applied, but then you look at the odds of being male. Vitamin D protects you against being male, and it protects you against being older. It's fantastic. And then so the papers were retracted. It's an effort to get anything retracted, but these papers got retracted. The authors then developed a new method which still has problems. They now say now it has no protection. I mean, this paper would not published in Lancet Diabetes and Endocrinology if they'd submitted a null paper. They wouldn't publish it. And they then said, well, it is possible for observational association. It is a logical impossibility, which it is. But logical impossibility is a very high bar to come down. Something has to be really seriously fucked up to be logically impossible. But many of these methods are basically black box. As I say, not even the developers don't really understand what they're doing. So what's to be done? Journals shouldn't send out our findings without something else, triangulation. Reviewers shouldn't review them. Journals shouldn't send out, should only send out papers that have filled out the Strobe MR checklist. This is where we can find the Strobe MR statement. One of the checklist things is that you have to say you've done is you have discussed the plausibility of gene environment equivalence. If people actually had to do that, then they wouldn't be submitting papers on gut microbiome, erectile dysfunction or breakfast skipping. So the real thing is that you can fill out this checklist, but often they're obviously getting filled out, but no one's actually, the editors and the reviewers aren't actually looking at them because they say, yes, we've done that, but they haven't. And methods should be more rigorously evaluated before implementation, including through use of negative and positive controls, which I'm very fond of negative controls. I think that's actually very powerful. So what it should become, so a bit slightly more upbeat for 8 minutes, and then I'll finish, honestly. So it should be presented within the triangulation of evidence framework. So MR, if done properly, thoughtfully, where gene environment equivalence is reasonable, then it can produce evidence which is worth putting into a framework where you're combining it with other evidence and orthogonal approaches which are on those questions. So you shouldn't just be MR papers, it shouldn't be just on their own, it should just be some. And some random, it sounds on some of these things are literally random outcome, random exposure, submit your paper. So triangulation is good. Absolutely, the one thing we really must do is reconnect the biology and cease to be just a mechanical application of instrumental variable analysis, which takes, I would say, takes you 5 minutes. So even if you're doing it yourself rather than paying someone else to do it. And the most influential current MR studies are either never published because they are being done by the pharmaceutical industry. And the pharmaceutical industry use this method. Usually they would not these days, they would try to go to randomized controlled trials for anything which could be genetically proxied if there wasn't genetic support for what they're looking at. And this was now last year, it's not 2026, this is in 2025. These are papers that appeared in the first 7 weeks of 2025 in Nature Genetics. Now, one of them has Mendelian randomization in its title, but all of the others, all of these contain Mendelian randomization as part of the follow-up of GWASs, etc. Nature will not fill in scope guidelines by the way. But that's so MR is appearing, the influential MR is appearing in a broader framework. It should contribute to the collection of appropriate data because where MR can be really powerful is looking at disease progression. Because that would speak directly to therapeutics. But think back to the American college. If you're looking at disease progression, you are stratifying on having the disease. You are automatically introducing collider bias. So if you're going to do Mendelian randomization on disease progression, then you need to use methods which can at least do sensitivity analyses on the effects of collider bias, etc. But so clinical cohorts and in fact the best data you can get off actually from randomized trials when measures have been taken on people at standardized times. That could be incredibly powerful for therapeutics. We applied to questions of natural selection. So I pointed out that natural selection is a subcategory of Mendelian randomization, and possibly my favorite piece of work I've been involved in the last few years was actually showing that everything that you've read in textbooks about lactase persistence is not wrong. You can actually use MR principles to look at what the phenotype is that's being selected, and it certainly isn't greater milk intake. We applied to non-human life forms. Again, things that economically matter, like pharmaceutical development and the amount of milk that cows produce, are being studied by using Mendelian randomization to look at economically important outcomes and rice. So there's a lot of work going on in this domain, and we've just established the Mr. Mouse Consortium, which is a consortium of people who do use the diversity outbred mice lines and have the same lumen chip data, and you can get vast numbers with fantastic phenotyping and use Mendelian randomization within mice. These are actually rats, not mice, but this is I saw this at the Lee Miller exhibition and I liked it, so that's going to be our logo for Mr. Mouse, even though it's actually rats. And you can. This is actually going. I'll skip over this, but you can use the Mendelian randomization principles to look at in vivo phenotypes and how whether they translate to in vitro phenotypes and translate between mice and humans, for example. So there's lots of stuff going on with MR principles, which is the biology, using the principles for biological investigation. And be used to understand how confounding generates robust and generalizable non-causal associations. One thing about when there's a very strong effect, like HDL cholesterol is inversely related to coronary heart disease, whatever is underlying that must be really important. So working out why you've got the wrong answer, so like the CRP, you can show that BMI, smoking, etc., what the confounders are. You can use MR to look at the confound. This is some work by Gus Hamilton, who was looking at HDL cholesterol, especially the small fraction of HDL cholesterol relates to lower risk of sepsis and sepsis death. That's what you see observationally in UK buyback. But if you do MR of this, yeah, it looks pretty null. But if you look at one factor, the IL-6 receptor variant, then that influences sepsis death. And this is what in COVID, of course, this was the genetic variant was shown to relate to lower COVID mortality, and that's what led to or fed into the fact that tocilizumab IL-6 receptor blockade was used for treating COVID patients, and maybe it should be used for treating sepsis patients. But the point of this is that you can actually show that this generates, just this one factor alone generates 50% of the observational association between HDL and sepsis onset and sepsis mortality. So actually understanding what the confounding is, actually understanding what those factors are, can be a very powerful way into working out causal networks. And I'll finish with an advert for this that came out last week. This is the genotype map project, whereby you can get this has been massively updated version of with a lot of molecular phenotype data, which can be used by our website and our package. And you can upload your own phenotype data into this and then use that to integrate it with the vast array of data. And it's starting to introduce sequence data, CMV, copy number variation, loss of function variants, etc. So just to revisit vertical and horizontal phylartropy with molecular phenotypes, much phylartropy is through downstream effect of a single molecular phenotype, which renders it as vertical phylartropy. But with the upstream phenotype, it's been extremely difficult to identify. So if you were using this to actually make a causal inference about the upstream phenotype, which is having many effects in many directions, that would be great. But you don't know that phenotype, then you get in the wrong answer. But there are cases of true horizontal phylartropy. And this is what with the genotype phenotype map we're trying to get better evidence on this. And the issue I raised with sclerostin is thinking of the cell or tissue type specificity. And I mean, one bizarre thing really in the expression world is people meta-analyze across tissues. So you'll get genetic variants and cis variants, but also trans variants related to expression. And they will have very different effects in different tissues. When people say, fuck that, and just meta-analyze it, we'll get some effect out. Now that's then disastrous, because if you then try to do Steiger filtering, you get the wrong answer, because your it could be that your genetic variant is explaining more of the variance in some outcome factor than it does in the exposure. But if you've meta-analyzed exposures across 19 tissues which aren't a tissue of relevance, then you'll just wipe out the signal, which will tell you that it's spurious. So you've got to think. The use of thinking about how you need to relate the causal processes actually to the cells and tissue types of importance. With the genotype phenotype map, trying to automate performance of bidirectional Steiger across all phenotypes, to identify implausible zones, propose implementation to flag those up, biological annotation of all steps of the MR process, highlighting suspicion in pathways under detailed interrogation, and MR model selection in relation to the properties of networks, analyze hopefully things to come, and naturally including positive and negative control analysis and propagating the results of these through the data network. So that's the paper. These are the people who did all the work. And this is one review on Mendelian randomization. If you're interested in the technicalities and the methods, if you rather trying to rant about how terrible some of the stuff is, this is a rant. or in evidence triangulation, or view of evidence triangulation. And then finally, if you've got someone, there's someone in your life you don't like very much, but you've got to buy them, you know, birthday present or Christmas present, I'd recommend this book as the ideal gift for someone that you don't like very much. Thanks very much.
Thank you very much, George. That was brilliant. You started by saying this isn't anything to do with the Institute, the Dementia Research Institute, but actually it's everything to do, both in the scientific framing and the need for rigor, and how you can use correct methodology to infer elements of biology that could then drive experiments and ultimately clinical trials, but the correct clinical trials. Questions? Will, and then our friend from Dundee. Will?
So if we think about some like dementia, people that's happening over the life.
Yeah, just speak up. Will.
If we think about some like dementia over the life, should we be trying to dissect these by looking at different populations, children populations, middle-aged populations, or is there some clever way of doing it?
No, I mean, I think that's a really good point. I mean, one issue, in dementia was the early finding from Iceland of a very clear, genetic influence, which was druggable, and then the molecular trial, the trials failed, and that might be because the process is just, as I said. The MR looks, you're thinking about the genetic variant appears to affect right the way across life, but if the genetic variant is influencing something during development, which then you're not going to change it by drugging it or by getting into a stage. For some phenotypes, you can separate them quite pretty nicely with Mendelian randomization about phenotypes at different stages of life. So that's true of adiposity, for example. So peak puberty, larger body size, peak puberty protects quite strongly against breast cancer risk, and the adulthood BMI effects are critically non-causal. So that it is possible, but you'd always want to go back to the genetic variants and actually look at how they influence things. Actually what their phenotypic associations are right the way across life. So birth cohorts like ASPA, etc., can be incredibly valuable for that.
Okay, thank you. Miracle?
In my own field of QI studies, they don't tend to really define all the variant or prove that the variant is either the gene that they assign or has a functional effect. Most of the gene variants are non-coding. In terms of your approach, is implicit in that you have to understand whether the variant is actually functional and is doing what is thought. And if you can't use QBAS list, you have to use clinical variants around characterized and functionally understood. Is that correct?
So I mean, I think you get the strongest inference when you understand variants, definitely, and the strongest inference when you can have when you know how the variants affect and then differ between across subgroups. When I talked about the various interaction effects, that's definitely the most powerful. If you get in situations where you have basically an anonymous variant and you don't know, but it does relate strongly to your phenotype of interest, then what we've been trying out and we showed that it works in the case of CRP, for example. If you Steiger filter against everything, if the genetic variant has a larger explains more of the variance in exposure you're interested in than anything else, then it doesn't mean it's going to be true, but it would make it that would be much less likely to be just entirely spurious than it is if you just go ahead and do the absolutely standard Mendelian randomization. So I think that's why databases like GeneSat, PhenoSat, Map can be incredibly valuable, because you can look to see if you look across a huge number of phenotypes, including molecular phenotypes, and the variant that you think is you're trying to use as a variant for exposure. If you then discover that variant relates to a greater variance in many other things, then you don't want to use it.
Thank you. Joanna?
Expressed in the analysis, so whether you worry about that so much because it's relatively minor here.
Yeah, I mean that's sort of interesting notion. I mean, you don't want to stratify on it. Methylation is a sort of phenotypic. You don't want to stratify on something which is phenotypic because that's very likely that other things will be influencing that. But you would know that genetic variant across on average, across the group is producing a robust on average difference in the exposure that you're interested in. That's you want to use that without actually taking into account the other phenotypes.
Anything else? Anyone else?